afternoon, dear Ms. Delvenne. Hereby I open this academic ceremony in which Ms. Aurore Delvenne, I hope I pronounced it correctly, will defend the academic thesis, her academic thesis, entitled CSF Proteomic Signature in Alzheimer's Disease across Amyloid and Tau Biomarkers Subgroups. May I invite you to present a summary of your studies in about 15 minutes and the conclusion of your thesis. And I'd like to give you the word.
Thank you. Dear Prorector, dear member of the Corona, dear colleagues, family, and friends, thank you very much for your presence at my public defense. Within the next 15 minutes, I will tell you more about my thesis entitled Cerebrospinal Fluid Proteomic Signature in Alzheimer's Disease across Amyloid and Tau Biomarker Subgroups. Alzheimer's disease is the most common form of dementia. It leads to a gradual decline in multiple cognitive function, including memory loss, which ultimately interfere with daily life activities. Due to the worldwide aging population, the number of individuals with Alzheimer's disease is increasing every year. There are currently no treatment to stop or reverse the disease. This is partly because we still don't fully understand the mechanism occurring in the disease. The human brain consists of billions of cells. Here you can see a healthy brain cell and a closer look at its internal structure. In Alzheimer's disease, two proteins, amyloid and tau, start to accumulate abnormally in the brain. Amyloid, here in pink, accumulates around brain cells. Tau protein, here in red, which normally help the brain cells to work properly, start to accumulate abnormally inside the cells and form tangles. This accumulation of amyloid and tau cause the brain cells to stop working properly and die, a process called neurodegeneration. The human brain consists not only of billions of cells, but also of billions of proteins that facilitate the communication between cells. When brain cells are damaged, protein levels change as the brain tries to compensate for those damages. We can measure those changes in protein level using a technique called proteomics. We can either perform a lumbar puncture, a procedure in which we collect a small amount of brain fluid from around the spine. The brain fluid is produced in the brain by a small structure called the choroid plexus, here in red, and circulates through the brain and the spine. With proteomics, we can quantify the levels of proteins in that fluid. We can also extract brain tissue and quantify the levels of proteins in that tissue. Nowadays, not only we have the technologies to measure thousands of proteins in the brain, but we can also assess the presence of pathologies in the brain. Indeed, amyloid pathology can be assessed through a lumbar puncture, as well as tau pathology. Neurodegeneration can be assessed through a lumbar puncture or alternatively via an MRI scan. By measuring these markers, we can classify people into subgroups. Individuals with no amyloid pathology, no tau pathology, and no neurodegeneration are considered as not having Alzheimer's disease. Individuals with amyloid pathology, whether or not they have tau pathology and or neurodegeneration, are considered as being part of the Alzheimer's continuum. Indeed, amyloid pathology is the main hallmark to define individuals as having Alzheimer's disease. Yet, there are some individuals that do not have amyloid pathology, but have tau pathology and or neurodegeneration. And those are classified under suspected non-Alzheimer's disease pathophysiology, or SNAP. Alzheimer's disease is characterized by the presence of pathologies in the brain, and those pathologies might lead to cognitive decline. Therefore, Alzheimer's disease progresses through different clinical stages. Individuals with normal cognition perform normally on cognitive tests. Individuals with mild cognitive impairment have noticeable cognitive decline, but daily life activities remain largely unaffected. The dementia stage is characterized by significant behavioral and cognitive changes which interfere with daily life activities. So there is an urgent need for new treatment for Alzheimer's disease. In order to develop this new treatment, we need to better understand the mechanism occurring in the brain in patients with Alzheimer's disease beyond amyloid, tau, and neurodegeneration. One way to gain insight into those mechanisms is the use of proteomics, and this is the approach we have taken in this thesis. So the first aim of this thesis was to better characterize individuals with suspected non-Alzheimer's disease pathophysiology, or SNAP. So those are individuals with tau pathology and or neurodegeneration, but without amyloid pathology. And we wanted to investigate their clinical characteristics as well as the mechanism occurring in their brain. So first, looking at the clinical characteristic, we performed a review of the current literature on SNAP, and we combined findings of around 63 papers in a meta-analysis. We found that SNAP was common in individuals with normal cognition and mild cognitive impairment, but less common in individuals with dementia. Over time, individuals with SNAP and normal cognition showed a relative cognitive stability, and they did not usually progress to the dementia stage. However, individuals with SNAP and mild cognitive impairment showed cognitive decline and progressed more often to the dementia stage. In order to investigate the mechanism, we performed lumbar puncture and we collected brain fluid, and we quantified the proteins in that brain fluid. This was part of an international study in which we included around 450 participants, and we quantified more than 3,000 proteins. So here we define SNAP as individual with tau pathology, but without amyloid pathology, and we first looked at individuals with normal cognition and SNAP, and we compared them with healthy participants defined as individuals with normal cognition and no amyloid and tau pathologies. We performed the same analysis in individuals with mild cognitive impairment and SNAP, again comparing them with healthy participants. We found that in individuals with normal cognition and SNAP, there were mainly increased levels of proteins compared to the healthy participant, while individuals with mild cognitive impairment and SNAP showed mainly decreased levels of proteins compared to healthy participants. Looking at the mechanism associated with those proteins, we found that there were also differences between normal cognition and mild cognitive impairment. Altogether, this suggests that SNAP represents distinct disease entities across the cognitive spectrum with different mechanisms occurring in the brain. The second aim was to investigate the neurodegeneration markers. So there are several ways to measure neurodegeneration. One way is to measure a protein called neurogranin, which is located inside brain cells in the postsynaptic area. Another way is to measure another protein called neurofilament light, which is located inside brain cells but on the axonal part. When brain cells are damaged, those proteins are released into the brain fluid and can be measured through a lumbar puncture. Another way to assess neurodegeneration is to measure the volume of the hippocampus through an MRI scan. The hippocampus is one of the first regions to show atrophy in Alzheimer's disease. Although all those markers are classified under neurodegeneration, it is still unclear whether they reflect the same mechanism and if they can really be used interchangeably as indicators of neurodegeneration. To address that question, we again looked at the levels of proteins in the brain fluid, and we included here about 400 participants. We compared individuals with Alzheimer's disease and neurodegeneration with those without neurodegeneration, and we performed the same analysis for the 3 distinct neurodegeneration markers. We found that those 3 markers represent distinct mechanisms and should not be used interchangeably as indicator of neurodegeneration. More specifically, individuals with neurodegeneration assessed by neurogranin showed mainly increased levels of proteins, while individuals with neurodegeneration assessed by neurofilament light showed mainly decreased levels of proteins. Interestingly, changes in protein levels in individuals with neurodegeneration assessed by the volume of a hippocampus were minimal. Looking at the mechanism, we found that they were also different between the distinct definitions of neurodegeneration. Our third aim was to assess the potential involvement of the choroid plexus in Alzheimer's disease, more specifically related to amyloid pathology. So the choroid plexus is that small structure in red in the brain that produces brain fluid, but is also important for brain immunity and for the protein transport. So to answer that question, we looked at mice, and we extracted choroid plexus tissue from the brain of the mice, and we quantified the levels of proteins in that tissue. We compared the levels between mice with amyloid pathology and healthy mice without amyloid pathology. In the same mice, we also collected brain fluid and looked at the levels of proteins. In order to distinguish between the protein that were associated with the choroid plexus from the other in the brain fluid, we clustered them based on their levels of expression in the choroid plexus. We also collected brain fluid and looked at the protein level in humans and compared those results with the mice results. So first, looking at the results for the choroid plexus tissue, we found that mice with amyloid pathology showed a high number of dysregulated protein in choroid plexus tissue compared to the healthy mice. Those proteins were related to the dysfunction of the morphology and the function of the choroid plexus. The results of the brain fluid were similar, again suggesting an association between choroid plexus dysfunction and amyloid pathology. To determine if those findings were relevant to humans, we examined the protein level in the brain fluid of humans. And here we found increased levels of proteins highly expressed by the choroid plexus in individuals with amyloid pathology, but without tau pathology, compared to healthy controls. We performed the same analysis in individuals with both amyloid and tau pathology, and there we could not find a direct link with the choroid plexus dysfunction. So this suggests a specific interaction between choroid plexus dysfunction and amyloid pathology. Looking at the clinical stages, we found that the choroid plexus dysfunction was related to amyloid pathology only in individuals with normal cognition and mild cognitive impairment, suggested that changes in the choroid plexus occur early in the disease process. Looking at the mechanism, we found the same as in mouse, so impairment of the morphology and the functions of the choroid plexus. So in conclusion, we can say that suspected non-Alzheimer's disease pathophysiology, or SNAP, is a heterogeneous biomarker-based concept which represents distinct disease entities across the clinical spectrum. Different neurodegeneration biomarkers of Alzheimer's disease are associated with distinct mechanisms and should not be used interchangeably. Choroid plexus dysfunction is involved in the early stages of Alzheimer's disease pathogenesis, particularly in relation to amyloid pathology. And altogether, we found that proteomic is a powerful and valuable method for uncovering the underlying mechanism occurring in the brain in Alzheimer's disease and other neurodegenerative diseases. I would like to thank you all for your attention and give the word back to the prorector.
Thank you, Ms. Candidate, for your very nice and well-illustrated presentation. Compliments to this. We will, and also right on time, also compliment. Before we start with our opposition, we have 5 opponents. I will also briefly introduce the promotion team, that's what we do currently. We have, for the people in the audience, opposite of me is the promotion team consisting of 2 members. On my right side, for you also maybe the right side, it's Professor Vos. She is affiliated with the Department of Psychiatry and Neuropsychology. And on the left side is Professor Visser, and he is officially affiliated also with our university and is in the field of molecular epidemiology of Alzheimer's disease. And I'd like to thank them. We're going to start, and basically we follow the list here. And we start with the chairman of the assessment committee of the candidate, Professor van den Hove. He is professor in neuroepigenetics at our university, and I'd like to give him the word.
Thank you, Mr. Pro-Rector. First of all, I would like to compliment you with this very nice thesis. A lot of work, well written down, and was a pleasure to read. And also would like to extend my compliments to your promotion team. But of course, I'm mainly here to ask you some questions, and let's start maybe with a seemingly or potentially obvious question. If I look at chapters 3 and 4, there's a lot of overlap, and in your general discussion you also compare the findings between the different chapters. But my question would be, why didn't you put all these different groups in one and the same analysis? As you also illustrated in your thesis, it was done in different batches anyway, but eventually all the groups or all the individuals come from the same cohort. So why didn't you compare them directly in one and the same analysis?
Highly esteemed opponents, thank you for your compliments and your question. So indeed, we had the same research question for the 2 different chapters. And first, we investigated individual with mild cognitive impairment and SNAP. So we begin with that. And then we did the other analysis in individual with normal cognition. And we wanted to know, we were first interested in the mild cognitive impairment because they showed cognitive decline. And so we wanted really to compare them with the people with the Alzheimer's disease to show if they had a similar underlying pathology in order to know if the cognitive decline were related to similar underlying mechanism or not. So that's why here in that first study, we did not include the normal cognition. And but then at that time already, we were interested in normal cognition, and we performed also analysis in SNAP with normal cognition, and there we find a really different profile. But that was also a lot to include in one study, so that's why we put it in 2 different studies. But in the discussion of the second one, we compared them. And also as you said, the batch were different. So
no, I understand that choice, and sometimes you start with something and then later on you come up with another idea. But did you then, just for your own peace of mind, still put them in one analysis by yourself later on? And if not, why not?
Could you please repeat the question?
So I understand your reasoning, but did you then, you could still do this, eh? You could still all run them through one analysis. I saw that maybe the controls are not completely comparable in terms of the age, etc. So there's a slight discrepancy, but maybe if you pool them, things are still okay-ish. So did you not just, out of curiosity, run such an analysis?
Yeah, we ran it, but as the first paper was already published, we did not put it in the paper, but we ran it and we found the same distinction between the 2 as compared to the 2 different papers. Okay. So that's why we did not include it in the last one.
Yeah, no, thanks. That is clear. Then I will move to a slightly different topic. So it actually is on the mouse work in chapter 6. I was wondering there, if I understood your approach properly, you have then the transgenic animals at 2 different ages, and then you compare them to the corresponding wild-type animals. And I was wondering actually why you didn't run a 2-way ANOVA with age as a variable. At least it seems that you did not. Because of course with an NS5 per group and per time point, per age, you might lack power. So maybe some of your results are false positive, or you also might have false negative results. And if you were to at least do a two-way ANOVA, you would not really lose any information, but you might actually gain something by getting slightly more statistical power. So why didn't you put them one and the same model, if I may ask?
Yeah, so first we started with running them separately, and we found that at the protein level they were really different. So that's why at the end we did not put them together in one analysis. That would have increased for sure the power, but then we will have a global picture, but not age related. And we saw that there were some huge differences, at least at the protein level, not at the pathway level. So that's why we did not do it.
Okay, I think you could still pick up the things that are different, but also look at the commonality, but actually looking at the effect of one, the other, and then the interaction between the two. But I can understand why you went in that direction. Another slide, practical question there. So you describe how you obtain the CSF of those animals, and then you do transcardial perfusion. I think my colleague on the left will ask you something about the cells that you then wash away. But I would like to ask you, okay, how does it then work if you approach the choroid plexus and you isolate it? Is it clean? I mean, maybe the blood cells are gone, but yeah, I know I did many dissections myself, not of the choroid plexus, so that's why I'm really generally interested in your answer. But yeah, you always have contamination because there's always some other tissue that comes along or whatsoever. So to which extent are you sure that you're actually looking at the choroid plexus proteome and not at anything else?
Yeah, this is a good question. So the extraction of the choroid plexus in mice is really difficult because it's really small. So it was not done by me, it was done by a technician with a huge expertise in that. And so that's true that there might be contamination of other structure next to it. But as they were quite expert in that and that they performed the same method for multiple times, yeah, I expect that contamination will be low, but that's true that yeah, we might look into that in the future to see if there is really some contamination. But yeah, I don't really expect a lot. Okay,
on that same study, you worked with female mice. I think also somebody else will ask another question on that, but you then try to translate those findings to a human setting. And then, as you also illustrated in your presentation, you compared it to CSF data, etc. But why did you actually then not compare them only to female data, but to data acquired in both sexes?
Yeah, so in order to increase the group, we put them together because otherwise it's reduced by half, approximately, the groups. So, yeah. And also, we don't really know if what we found in mice, in female mice, would be the same as in, will be also sex related in the human. So that's why we prefer to pull all of them together. But that's something that needs to be done with a higher data set in order to distinguish between male and female. And also, we corrected for age in our analysis, for sex.
Yeah. If I may, a last short question. Yeah, it's nice that you stratify your groups and based on amyloid, tau, and neurodegeneration. Did you try cluster analysis if you ignore that, and would you end up with actually the same groups based on your proteomics profiles if you would ignore those markers?
Yeah, we did not try that, but that's an analysis that should be nice to do. And yeah, we will try that in the future, but yeah, we did not do that.
Well, I get the question you're short, and you the answer to this, I think, to your satisfaction. So I give the word back to the pro-rec. I'm also very pleased with your answers. Thank you.
Thank you. And indeed, it was perfectly short on both sides. The opposition will be now continued by Professor Martinez. She's professor in neuroinflammation and neuropsychiatric disorders, and I'd like to give her the word.
So, dear candidate, I compliment you with your thesis. I think a lot of work. Also, you had to do different methodologies and to integrate all together. So that's very, very remarkable. Also, my compliments to the promotion team because I think it's quite a diverse topic. Also, you study human samples and animals, which is a difficult sometimes to combine. So my question to you goes to chapter 6 regarding your animal study, and especially I'm very interested about the choroid plexus because, as you also mentioned, is important for immunity, and there are antigen-presenting cells presenting the choroid plexus, etc. And then I was, when reading your chapter, then coming back to the question of Professor van den Hove, you use female animals. Then I was wondering, and then you, in your statements, you want to do a patient-specific finding. So you want to have personalized treatment. Then coming back then to the study, I realized that you only look to the female. So you also mentioned about the limitations of the female, of using only females, but could you elaborate a little bit more in which aspects perhaps to use females might have a little bit biased your results? Because I think you found several pathways and you might know which pathways might be quite affected by sex. So could you elaborate about this a little bit?
Yeah. Highly esteemed opponents, thank you for your compliments and your question. So indeed, that was a really strong limitation in our study. Also, the groups were quite small, so it was not possible to include both mice sexes in our analysis. And indeed, it might have led to some sex-driven changes in the proteomic, because we know that in a female there are a lot of hormonal changes that happen and that can interfere with the proteomic profile. I think that's the main thing that could have led to changes in the proteomic profile.
Yeah, so for example, proteins might change because of sex, but also lipids change remarkably because of sex. So I think if you really want to have an answer, you really need to have both sexes. Now, coming back to the same question about the choroid plexus. So when you look to the function of the choroid plexus, when you have the choroid plexus are malfunctioning, as you, in your conclusion of your study, you claim that because of changes in proteins, there is a malfunctioning, a dysregulation of that part of the brain. But it is known already which changes you might expect regarding function on that. For example, do you know some changes that you might be able to measure in the animals due to malfunctioning of the choroid plexus?
Yes, I think immune changes can be measured because so the choroid plexus is a structure that allows brain cells to come into the brain, into the CSF, and so I think looking at those immune cells might be a first step. Otherwise, lipids might be also important because choroid plexus transport a lot of lipids. So I think. Yeah, so I was also more going into the changes in the volume of the CSF, or even if there is blockage of the CSF due to malfunctioning of the iron. So it would have been very interesting in your study to analyze those as well. But now coming back to this question, so you mentioned that your mice are perfused, right? So you perfuse the animal, so you don't have immune cells or blood into the brain when you collect the tissue. However, I was wondering, so that's very good to do when you work with mice and you want to have a pure tissue, but then you compare your analysis to human tissue which has not been perfused because they are post-mortem retrieved and you cannot perfuse it, of course. And then do you miss a control in your study that you might have to include in order to be able to conclude whether the findings that you have are they are masked perhaps by the fact that you perfuse your animals? Yes, I see your point, but I don't look at tissue in humans, so I'm not sure I understand correctly your question. Because I don't do tissue analysis, I only do CSF analysis. Okay, so you only do CSF analysis. In the case that you will be using the human tissue, so what will you need to do? Which control will you need? If I do analysis in the tissue of humans. Yes. And you want to compare it to the... Because you didn't... So now you say you only compare the blood analysis, but not the brain, but there is a reason for that. So why? Why you didn't go to the tissue? Yeah, we didn't go to the tissue because it's more difficult to have data on the tissue because choroid plexus is quite difficult to extract in human because you need to extract it directly after death, because otherwise there is changes in the protein composition of the tissue. So it's quite difficult, and also, to have those samples, it's
but one of the limitations would have been that you will need additional control in your animals of non-perfused animals to be able to compare to the non-perfused human tissue. Okay, that was just very interesting. So if I would like to go to chapter 4, where you have two different populations of individuals with different cognitive characteristics, and you have two remarkable groups, but then I didn't find in your chapter, perhaps I missed it, whether you have matched this for sex and age when you compare these two groups. So could you explain me about how you did the analysis regarding this aspect?
Yeah, so in chapter 4, so we corrected all analyses, all proteomic analysis for age and gender. We, in general, in whole chapter, we corrected for age and gender, as we know that between groups, especially with control, there are some age differences and also gender differences.
So, so the two groups were matched for sex and age, but they were mixed, right? So you had females and males together.
Yeah, so they were not matched, but they were just, we corrected in the analysis. We performed ANCOVA, corrected for age and gender.
Yeah, because I really think that if there are already naturally differences in the age of these groups, that perhaps this comparison is not very easy to perform when looking to these two different entities. And I think you should keep that in consideration when looking. So, I think I have my time. I don't know if there is still room for additional questions.
There's 1 minute for a short question.
Okay, so you also find antibodies. Could you elaborate which type of antibodies you see to be present in your analysis? Did you analyze the type of antibodies that you find? Because you mentioned antibodies as well as lipids are changed between, in general, in this proteomic analysis. But did you analyze which type of antibodies, which isotype of antibodies was changed?
No, we did not have a lot of data on antibodies, so we did not perform that, but that's something that's with now all those bigger immune panel that we can do with more targeted proteomics. That's something that, we might look at, but we didn't look really at it.
Mm-hmm. Thank you. So I give the voice back to the prorector.
Thank you very much. Then the opposition will be continued by Dr. Pichva. He was a member of your assessment committee, and he's affiliated with the Department of Psychiatry and Neuropsychology at our university, and I'd like to give him the word.
Thank you very much. Thank you very much for the very nice presentation, clearly explained what you have done over these years, and my compliment to your thesis and the nice studies that you have done to you and to your promotion team. I enjoyed reading your thesis very much. If you agree with me that you have used heavily pathway enrichment analysis to interpret the findings, which basically you are investigating or you are characterizing the ATN subtypes of, like, in individuals, not only in AD, right? Mm-hmm. So, but your interpretation of the findings are heavily dependent on the pathway enrichment analysis, right? I was wondering if you have given thought or some research that what are the bias and limitations of using this method?
Esteemed opponents, thank you for your compliment and your question. So indeed, all our analyses were driven by the pathway, and we did not look, we did look at the proteins, but not much, because here we do not correct for multiple testing. So we are not looking for a biomarker, but of course looking at the pathway with those gene ontology tools, they might not take into account all proteins. There are also some proteins that might not really be, when we do targeted analysis, that might not be significant anymore, other that might be. So it gave a general overview of the pathway, but some pathway might not be really implicated in the disease. So that's why usually in the conclusion we only included the top 3 or 5, depending of the study, the top 3 or 5 pathway.
Okay. If I understand correctly, you are saying that so when you limit yourself to the pathway, you limit yourself to the prior knowledge, and then you are not able, or basically you are not letting new discovery happen, yeah, when in your interpretation phase at least, right? But you also mentioned that you did not correct for multiple testing yourself, and you end up with relatively large number of proteins as a differentially expressed, right? And can you tell me that why didn't you do that?
Yes, because when we corrected for multiple testing, then the number of proteins were really limited. So then we could not find pathway, and yeah, usually multiple testing correction are used more for biomarker investigation, and here it was not the point. It was more to know the pathophysiology in general and not to come up with specific proteins to target or to measure to detect the disease or...
Okay. I have to disagree with you that the multiple testing correction is for the biomarker research. So I think the importance of that for the relevant proteins or genes or anything that we are identifying, that it's not a false discovery, while we want to invest a lot in the downstream analysis or even finding a target for treatment, it's very important to make sure that we are not investing on false discoveries, right? But I was wondering that because you could think of, okay, combining the p-values with some sort of fold change measures, so at least you take into account the magnitude of the changes and a little bit narrow down the thing. But okay, so you gave the answer. But then I'm looking at the volcano plots, and I see that the X bar is always labeled as log fold change. However, you perform ANOVA. I was wondering if you have calculated the fold change from the output of ANOVA and the post hoc, or if not, how do you think that it is equivalent to fold change, the statistics that you get from the post hoc of the ANOVA?
Yeah, each time we looked at the fold change, and could you please repeat your question?
So I'm saying that usually when we are using ANOVA or ANCOVA, right? So statistics that it gives us is F statistics, and then when you go for the post hoc Tukey's test or whatever post hoc that you for the paired comparison, then basically you are looking at the difference between the variance of means, and that is not representing fold change, right? But the volcano plots were labeled, so maybe it's just mislabeling.
Yeah, I think so.
Okay, so there was no calculation of the actual fold change in the proteins from the statistics.
We calculated the fold change based on the controls.
Based on the z-scores that you calculated, or like you normalized on the controls?
We normalized on the controls.
Okay.
Yeah.
So you believe that the statistics that now you get because of that normalization is equivalent to the fold change?
Yes.
More or less. Okay, nice. Another question is that when I'm looking at the pathways that you have identified in multiple comparisons that you have done, I see that the extracellular matrix is standing out. I was wondering that in this context of the heterogeneity in terms of the ATN and all the comparisons, since it's been repeated also in the upregulated, downregulated subsets that you have made, I was wondering that, first of all, if you can a little bit elaborate that what do you think that the role of these pathways or cell component is in the context of your research? Or have you looked into some of the proteins that they are involved and the direction of them, that how they differ between the groups comparisons?
Yes, so we did not directly look at the proteins themselves. I looked a bit at them, but that was not the focus. But the extracellular matrix could be involved differently in the different papers here, because extracellular matrix is important in the choroid plexus, and we saw in SNAP a lot of decreased proteins linked to the extracellular matrix, linked to the choroid plexus. And more on the AD, we found increased amount of proteins linked to the extracellular matrix, and that could be linked more to the synaptic plasticity, for example, in chapter 5, because there should be changes in the extracellular matrix to allow the plasticity of the neurons. So I think those are a bit different across the different chapters with different implications.
Yes, thank you. So getting back to my first question that I asked you about the bias of pathway analysis. So one was that like you're limited to the prior knowledge and the pathways that they are defined based on literature, etc. Another bias is like a bit methodological bias, because it is known that if you have a pathway or gene ontology with a large number of genes, the chance that you find overrepresentation from your identified list of genes on that is more often, right? So have you done anything to avoid this?
So in gene ontology, all pathways are corrected for multiple testing if they are corrected. So this is the only thing we have done to encounter that problem, but that's true that we are aware of that. But that's something that is difficult in proteomic study in general, I would say, to avoid.
I mean, like it's a general issue with this kind of analysis, but when you are looking into Panther, for example, that you have used, you can select that I'm interested only on the pathway that they have at least more than 10 genes and less than 2,000 genes. So you have this configuration that you can select to a little bit avoid this phenomenon.
Yeah.
Okay. So then in terms of the ECM, the extracellular matrix, there was a study, I think 2017 or something, that they have checked the association of 5 ECM proteins with the diagnosis and also the binary cutoff of A-beta 42. And they did not find any association between the 5 ECM proteins and these outcomes, so AD outcomes or CSF outcomes. But you are finding something, apparently. Can you tell me that in the context of precision medicine that you are describing in your thesis, that how is it justified? Is it like, okay, a little bit?
Yeah, I think now we are going to an era where we need to look more at individuals specifically, and there has been in Amsterdam a paper on subtypes. So I think this is the direction we need to go. And some individual might show a link between amyloid pathology, for example, and extracellular matrix, and some don't, and then the findings are not significant. But this is something that requires a big data set. So I think this is the direction we need to go, but yeah, it will take a bit of time.
I think it was the advantage of your study by stratifying the AD group to these ATN subtypes and then looking at this association. Now you find something, and that's basically you address the goal. Yeah, thank you very much for your answers.
Thank you very much. The opposition now will be continued by Professor De Wachter. She is professor in neuroscience at Maastricht University in Belgium, and I would like to thank her that she joins us today.
Thank you. Dear candidate, I would like to start with congratulating you with this outstanding work. I think it's really outstanding, as I say. It's reflected also in the publications. And it touches on a topic which is very important in the AD research, and it's studying the pathophysiological mechanisms and the pathophysiological mechanisms of AD in humans, and pushing beyond amyloid tau and neurodegeneration and trying to understand how the processes are involved and which other processes we can tackle in addition. So I think it's really an excellent, outstanding work. The work really focuses on analyzing biomarkers and using them for pathophysiological insights. So what I would like to ask you is, did you put all of this together in your head? And what is your favorite version of the amyloid cascade hypothesis? So you have amyloid, tau, neurodegeneration. You have many different processes in between, so I would be very eager to hear your version of it.
Highly esteemed opponents, thank you for your question and your compliments. Yeah, that's a hard question, I would say, because yeah, we found also different things between the different chapters. So I would still say that the amyloid hypothesis starts with amyloid, even though we don't see that in SNAP, but those are really a subset. So yeah, I would say that in the more general population, it starts still with amyloid pathology, following by tau pathology, and then neurodegeneration, because we saw in our ATN paper that most of the people that do not have tau pathology have only one or no abnormal marker of neurodegeneration. So I think it is in the same direction as the amyloid hypothesis. Yet there are some individuals that are SNAP, and it's difficult to know what they are from a pathological perspective. At autopsy, we know that 7 to 9% of those SNAP patients have actually amyloid pathology, so they might follow the amyloid hypothesis, but have a subthreshold biomarker. And also then we know that they have highly increased amyloid production. So maybe our cutoff are not sensitive enough. So I think the hypothesis, as it is now, it is quite a good hypothesis. I will go in that direction. But yeah, we should be careful of individual differences.
Mm. And you say so they don't have amyloid pathology, yet they have increased levels of A-beta. Do you think that other forms of A-beta could be the toxic culprits, and does this support this? And which types of amyloid, or can you elaborate a little bit on that? Yeah,
I'm not sure there is another type of amyloid that is really pathological here, because we looked at the data set including multiple isoforms of amyloid. It's not published in the thesis, but we looked at that, and there we could not find really a significant type that was more increased than another, and that might lead to the pathology. So I would say that in SNAP it's really tau pathology, and this is something that we saw also on the proteomic profile, because we saw the same, at least in individual with normal cognition, the same profile between A-T plus and A plus T minus.
Mm-hmm. So you don't consider oligomeric amyloid peptides as possible?
Yeah, that's true that oligomeric peptides could be, but there are no way to measure them.
And then your idea on what are the processes that are induced by A-beta, by tau. I hear you talking about synaptic processes and so on. So what would there be your favorite hypothesis?
Yeah, so I think the choroid plexus plays a major role in both amyloid and tau pathology. And we saw a different profile in individual with amyloid only and with tau only. And also in previous studies on subtype, they find a subtype related to the choroid plexus. So I think the choroid plexus might be the trigger of the disease for both amyloid and tau. Synaptic plasticity is also important, more on the early stage, with mainly hyperplasticity to cope with the neuronal damages, and then it decreases.
Yeah, and indeed, probably also some other neuronal processes and glial processes will be involved as well. I want to extend a little bit on it has been mentioned before, some analysis has been done in CSF, some analysis can be done in brain tissue. So the proteins that you pick up in CSF, they derive from neurons in glial cells. They are intracellular proteins, right? Mm-hmm. How do they end up in the CSF? Is this always by damage of the cell? Are there active processes? Are they secreted? Are they released? So The picture that you get in the CSF, or maybe they aggregate, and you have less in the CSF and more in the CSF. So the picture that you get in the CSF, how reflective is that of processes occurring in the brain?
Yeah, this is a good point. Yeah, it's difficult to know because there are multiple things that could interfere with the protein production, the protein transport. There might be also more epigenetic changes. So it's difficult to know what really leads to that. And I think by doing multi-omics and integrate a lot of different technique, more genetic, more proteins, more also tissue analysis, we can better know that, but at that stage that's difficult to know.
Mm-hmm. Yet there is a big advantage in measuring biofluids, which is quite obvious and very interesting. I would like to go with you to page 141 of your thesis, where you have a table.
I just have to ask a short question. Okay.
So the link between amyloid and tau. So there you have a stronger correlation in A plus T minus mice, humans, A plus T minus compared to A plus T plus. So what do you think about this? The correlation between p-tau and A beta 42 over A beta 40. So indicating.
Yeah, so
So what is your interpretation of those data, the correlation between differences in p-tau and altered A beta?
Yeah, so we saw that the correlation were, there was no correlation with A beta 42 in on individuals that have amyloid pathology.
Yeah, I'm talking about ratio 40-40, so representing plaques.
Yeah. So there we see a negative correlation.
Well, do you think the both are linked?
No,
Yes
we
or
have
no?
to stop it now, okay? Thank you. I'm sorry, because we get question after question. That's not the case. And we still have a final opponent who also is rather eager to join here the discussion. I would like to welcome Dr. Kuiperet. She's affiliated with the Department of Neurochemistry and Molecular Biology of the Radboud University Medical Center in Nijmegen, and I'd also like to thank her that she joins today, and I give her the word.
Yeah, thank you. Dear candidate, first of all, I want to congratulate you with a nice thesis about signatures in the CSF of AD-related patients groups. And in this, I also want to extend a congratulations to your supervision team. And I think you've gathered a lot of information that is very important to know more about the pathophysiological mechanisms that's playing a role in underlying Alzheimer's disease. So you did proteomic studies, and you found a lot of upregulated and downregulated proteins, and then you performed pathway analysis. And in these analyses, you separated the upregulated and the downregulated protein. And my first question is, why did you make that choice?
Esteemed opponents, thank you for your question and your compliments. So indeed, that's something that we have done, but that's sometimes is done in the literature, but not always. Sometimes they put all proteins together. As our simple size and the number of proteins allow us to distinguish between the two, we prefer to distinguish, because then we could exactly know in what direction the pathway were going. So if we saw more hyper or hypoplasticity and so on, but that's true that all proteins interact with each other. So one dysregulated proteins could lead to the upregulation of another. So that's a good point that we looked at, but in all paper, finally we decided to separate the protein, because putting them together supported also the findings.
Yeah, I'd like you to think along with me a bit further, because how does it work in a pathway? Do you have consecutive events that are going in one direction always, or can it be different? And another thing is, you look in the CSF, and it's supposed to be a reflection of the processes that are going on in the brain. But for instance, if you look at Alzheimer's disease, as you know, you have aggregation of the accumulation of the amyloid beta and the tau protein. But if you look at the CSF, the tau protein is increased in the CSF and amyloid beta protein is downregulated. So then again, I would like to ask you, is it indeed, if you want to find pathophysiological mechanisms, the best way to look at the up and down regulated proteins separately?
Yeah, I think that's still interesting to look at them separately, because by looking at them separately, you can see what differences there are, because amyloid is decreased in the CSF, tau is increased, and it can, by measuring that, we can say that the amyloid is aggregating inside the cells, while the tau aggregates inside the cells, and then they go to the CSF when the neurons die. So I think this is important to look at them separately, to see that.
Okay, yes, thank you. So a bit further on the pathway analysis, can you help me a bit on how it works, the analysis? So what does it make that the pathway is dysregulated? So for instance, is there a certain number of proteins that were changed that have to be associated with a certain pathway, or does the degree of increase or decrease of a protein, the significance of it matter? Can you explain a bit more about how that works?
Yeah, so the significance of the proteins do not matter in a pathway analysis. It's just the number of proteins that are globally associated with that pathway compared to the number of proteins we found in our analysis. Then there are some FDR correction, and it needs to be, the ratio needs to be higher than a certain degree.
Yeah, so if you, for instance, in chapter 6, you have figure 4 on page 184. So you looked at correct plexus, the mouse tissue, the mouse CSF, and the human CSF, and you compare that. And if I understand it correctly, on the left-hand side you have the pathways, and on the right-hand side more or less the processes that they are linked to. And you see for some processes that there are multiple pathways linked to that, and another one, immune, for instance, there's only one pathway linked to that. Can you explain a bit what I can read in this figure, and which pathways are more important? Is there something to say about that?
Yes, so here we clustered manually the different subpathways into broader categories. So for example, here, extracellular matrix. So this is something that was done, as I said, manually. So this is not something that is done by a specific tool. And here you see, for example, that they don't always go in the same direction. You have some decreased, some increased. So it's a global image that I perform myself based on the broad category, and looking at the single pathway give insight on what exactly is happening in the category I made myself.
Okay, then you mentioned also in this chapter and other chapters as well that it's important to know about the underlying pathophysiological mechanisms, also to be able to identify novel treatment options as well. So if you look, for instance, at this figure in chapter 6, what would be your advice? Which pathways should be targeted based on your data?
Yeah, that's a good point. It's difficult to say it. I would say that it's more the correct plexus in general that should be targeted, and and not really a specific pathway. But here, as really the most significant pathway were linked with the extracellular matrix, I would say that we should trigger that. But yeah, I think it's difficult to really trigger that specific point, so I would say more trigger the general functioning of the great plexus.
Yeah, but it's difficult to develop a treatment based on more a broad pathway. So can you suggest some more other studies that you have to do to get a better idea about it?
Yes, so you need to do...
Dear candidate, you are allowed to answer the question, but you also can say, I don't stop here. I prefer to stop. It's up to you.
I can answer quickly. So I think we need to do other analyses like histopathological analysis and so on to really see what is really happening in the tissue itself.
Thank you very much. Dear Ms. Candidate, dear Ms. Delvenne, the degree committee should mention that the time appointed for defending your thesis is now over, has passed. The degree committee will now withdraw to discuss the quality of your thesis and in particular the way you have defended your thesis this afternoon. And I request that you and your company will await the results of our deliberations and our return to the aula. Thank you very much.
Please all rise.
The PhD defense has now ended. The degree committee will debate the candidate's performance behind closed doors. This process usually takes about 10 minutes.
Long road to the south side. 7 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and die. On the line, I know you know that I can't read your mind. Years can't fly by now, don't waste all your time. Cause I'll go the extra mile. Long road to the south side, 7 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and die.
On the dash, got my laces tied. Long road, I don't waste no time. Break rules because fate decides. With the team and we chase the light. I make a move, fall down, shake it off. I hate to lose, bad branch, break it off. No room for negativity, praise and love. Prepare for deep part cause we're taking off.
Take the mileage, traveling so far from home. Gathering knowledge, other connections. I put the time in to get everything I ever hoped for. It's a challenge and we all in. It's a better future that we go for. And even though these nights seem long, I know that I am never on my own. Know you won the battle once you grow. South side, 10 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and die. Long road to the south side, 10 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and die. line. I know you know that I can't read your mind. Years can't fly by now, don't waste all your time. 'Cause I'll go the extra mile.
Dear Ms. Candidate, dear Ms. Delvenne, the degree committee here present has assessed not so much the quality of your thesis that was approved, but in particular the way you have defended your thesis this afternoon. In view of its positive verdict and taking into account your previous qualifications, the degree committee has decided to grant you officially the degree of doctor. Professor Visser is authorized to confer upon you this academic distinction in accordance with Dutch university custom. And therefore I invite your supervisor now to take the floor.
Please all rise.
Congratulations. So, do you promise to work in...
Please use the microphone.
Do you promise to work in accordance with the principles of scientific integrity at all times, to be careful and honest, transparent, independent and responsible?
Yes, I promise.
Okay. By the authority vested in us by law and in conformity with the decision of the committee here present, I hereby confer upon you, Aurore Chloé Marie Jean Delvenne, the degree of doctor and grant you all rights attached by custom and law. As evidence of this, I now present you the decree, certified, signed by the rector, the secretary and other members of the committee, and affixed with the official seal of the university.
I give the word now to Dr. Vos, who also gives you an additional degree.
On behalf of the Research Institute of Mens, I'm entitled to hand out his official certificate. The owner of the certificate has received training in a broad range of academic skills and has gained experience that is relevant for a successful career as an independent scholar.
And last but not least, there will be another certificate, a third certificate. And now we're going to give the word to Professor De Wachter, who will be here as the Euron representative.
On behalf of Euron, the European Graduate School of Neuroscience, I'm also entitled to hand out the official Euron certificate. Euron is a training network of 8 universities in the Netherlands, Belgium, Germany, France, and Luxembourg. The certificate is awarded to PhD graduates that have participated in multiple courses and conferences of the Euron program, that have collaborated and worked in a foreign research group, and that have published their work in international research journals.
And now I give finally back the words to Dr. Vos, who is going to speak the laudatio for the new young doctor.
Esteemed Dr. Delvenne, dear Aurore, congratulations on achieving this remarkable milestone by earning your doctorate. It's an honor to celebrate this success with you. You've produced a wonderful dissertation with all your papers published in prestigious journals. Following today's excellent defense, we have every reason to be proud. In January 2020, we offered a PhD position for CSF proteomics project focused on Alzheimer's disease. Your background in biomedical sciences and expertise in lab work brought a unique skill set to our team. We believed it could be of great fit to the project as we explored the brain's underlying processes, even though we couldn't offer traditional laboratory work, and large-scale proteomics was a completely novel field for me, with only preliminary results at that time. We took the risk, and you embraced the proteomics challenge within a team with a more clinical background. The rest is history. Your thesis is an excellent example of how various findings have led to novel studies that nicely came together throughout your PhD journey. You conducted a meta-analysis on SNP prevalence, based on all available studies to date. And using CSF proteomics, you have been one of the first to study the underlying processes in persons with SNAP across the clinical spectrum. This work paved the way to further studies on the involvement of the choroid plexus in SNAP and AD, as well as in mice, and for eventually examining the underlying brain process also associated with different neurodegenerative biomarkers profiles. It's been a pleasure to witness this process unfold as a result of very hard work. Through large-scale international collaborations, you perform proteomics data analysis, for which you effortlessly customized R scripts and found every time a new way to visualize your results in pretty graphs. You have also spent several months with Prof. van den Broeke and our colleagues at VIB Ghent to compare our human results of choroid plexus findings with mice. Although the start of the internship was challenging, it ultimately became a great experience, a great learning experience. You ended up doing some lab work eventually in mice, and this resulted in a beautiful translational research project with exciting results for the Alzheimer's field. You presented several of your findings at leading international conferences. Additionally, you supervised multiple biomedical students in completing their bachelor thesis. You slightly improved your Dutch skills. And not to be overlooked, on the side you also completed the second thesis of over 100 pages as part of another master in management in Liège. Who would do that? Moving from 100 pages to 100 slides, how do you manage to include 50 slides in a 10-minute presentation? That said, there were some bumps on the road as well. Just a few months after you started, COVID hit. The CSF lab analysis, they faced significant delays. The timing of your internship abroad, in your home country, remained uncertain for a very long time, affecting various aspects such as your teaching experience, course planning, and also the timelines on your data analysis. Yet, you never gave up and consistently demonstrated your commitment. Even when Verviers, where you live, was flooded and you were left without electricity, you kept on going. Early on, I learned that your name Aurore means daybreak, sunrise, and is often associated with the arrival of light, tranquility, and new beginnings. Well, this is exactly what you have brought to the project and the team. I'm glad we have got the opportunity to get to know you better throughout the years. Initially, you were reserved and a modest person, but you gradually opened up to your colleagues. Your motivation, enthusiasm, attention for detail, and perseverance in your work define you. These skills appear to be also reflected in your free time activities, such as your Barbie Girl karaoke performances and impressive Halloween outfits. My Belgian colleague and proteomics partner in the team, until this summer, for the past 4.5 years, my working week would typically begin with a meeting with you. And what a joy that has been. Beyond the usual weekend updates, I greatly appreciated our long discussions on the role of multiple proteins, the generation of new hypotheses around our results, and the interpretation of those visually appealing graphs. Our meetings would always provide fresh ideas to further explore, creating the best vibe to kick off a working week. Dear Aurore, it has been wonderful to see you develop as a researcher and become a real proteomics expert. I'm grateful to have had the opportunity to supervise you throughout this project, this journey. Congratulations on this achievement, and I extend my best wishes on behalf of the other half of the promotion team. I wish you all the best in your new role at ACL as a postdoc on proteomics. You will continue on our proteomics CSF project, and I hope that we can continue to collaborate. Je voudrais finir par féliciter aussi ta famille et Laurent, and now it's time to celebrate.
Very well. Dear young doctor, dear Dr. Delvenne, how does that sound? Feels good?
Good.
Good. Also on behalf of Maastricht University and the Board of Deans of our university, I would like to congratulate you with this, what we call ultimate academic title, which you can achieve. I would like to also congratulate your promotion team who helped you in this last 4 years to achieve this here. I would also like to thank the members of the Corona who raised a lot of questions to you in a very broad spectrum, and you answered them in a very elegant way. Thank you very much. I would like to congratulate your family here, who came all the way from Verviers, I suppose. Yes? So I'm not going to speak any French. I was hesitating, but I'm not doing. So basically, thank you also for joining us. And I would like also to thank the members here of the audience who likes, who entered here today and joined us for this festivity. And by that, I'm officially going to close the ceremony. For this, I have to use my hammer, which you haven't seen, but it's right here. And I close the ceremony. How do we proceed now?